Welcome to a fascinating exploration of the human body's intricate communication network. Today we are diving deep into a subject that might seem distant from your primary field, yet is fundamentally connected to it, the nervous system. As future food safety officers, your primary mission is to protect public health from the dangers lurking in our food supply. You will become experts in microbiology, sanitation, and regulatory frameworks. But understanding how the human body reacts to the very threats you are working to prevent provides a powerful and essential context for your work. It elevates your knowledge from a set of rules to a profound appreciation of human physiology and vulnerability, making you more effective guardians of public well-being. Why should a specialist in food safety care about nerve impulses? The answer lies in the body's immediate and often dramatic response to contamination. When an individual consumes food tainted with pathogens or toxins, it is the nervous system that sounds the first alarm. It orchestrates the body's defense mechanisms, from the initial feelings of nausea to the more severe symptoms that signal a serious health crisis. This rapid communication system is what differentiates a mild discomfort from a life-threatening emergency. Knowing how these signals travel and how quickly the body can react gives you a deeper understanding of the critical importance of preventing contamination before it ever has a chance to trigger these powerful biological responses. Think of the nervous system as the body's ultimate surveillance and emergency response team. It is constantly monitoring the internal environment, processing information, and executing commands with lightning speed. The principles of nerve impulse conduction are the very language of this system. By learning this language, you gain insight into the mechanisms behind symptoms like vomiting, diarrhea, or muscle paralysis, which are classic signs of foodborne illness. This knowledge is not just academic, it is a bridge that connects your professional duties to the real-world human impact of food safety failures, reinforcing the gravity and importance of your future role in the industry. This journey into the nervous system will begin with the basics, building a solid foundation for your understanding. We'll explore how a single nerve cell can generate and transmit a signal, a process that underpins every thought, sensation, and reaction you experience. This is not just a lesson in biology, it is a window into the very essence of how our bodies perceive and interact with the world, including the food we eat. Grasping these core concepts will equip you with a more holistic perspective on food safety, one that respects the delicate and sophisticated biological systems you are sworn to protect from harm. At the very heart of the nervous system's incredible capabilities lies a single specialized cell, the neuron. This is the fundamental unit of communication, the building block from which the entire complex network is constructed. Think of the nervous system as a vast and intricate electrical grid that powers a bustling city. The neurons are the individual wires, each one responsible for carrying vital messages from one point to another. Without these wires, the city would fall silent and dark. Similarly, without neurons, our bodies would be unable to process information, coordinate actions, or even maintain the basic functions necessary for survival, making them essential to our existence. Each neuron is a marvel of biological engineering, perfectly designed for its role as a high-speed messenger. It consists of several key parts that work in perfect harmony. The main cell body, or soma, acts as the neuron's command center, containing the nucleus and other essential organelles that keep the cell alive and functioning. Extending from the cell body are branch-like structures called dendrites, which act as antennas, receiving incoming signals from other neurons. A long, slender projection called the axon is the primary transmission cable, responsible for carrying the electrical signal away from the cell body and towards its destination, which could be another neuron, a muscle, or a gland. Understanding the structure of the neuron is the first step toward understanding how it works. The signals that travel along these cellular wires are not like the simple flow of electricity in a copper cable. Instead, they are sophisticated electrochemical events known as nerve impulses or action potentials. These impulses are the universal language of the nervous system, allowing for rapid and precise communication over both short and long distances within the body. It is this process of impulse conduction that allows you to instantly withdraw your hand from a hot surface or enables the brain to interpret the taste and texture of the food you are eating. Before we delve into the dynamic process of how these impulses are generated and propagated, it is crucial to appreciate the neuron as the cornerstone of this entire system. Every sensation, from the pleasure of a delicious meal to the discomfort of food poisoning, is translated into the language of these tiny cells. As we proceed, we will dissect this process, breaking it down into manageable stages. By the end, you will have a clear picture of how this fundamental biological mechanism works, providing you with a deeper appreciation for the physiological events that you, as food safety officers, strive to prevent every single day. To truly comprehend how the nervous system communicates, we must first become intimately familiar with the anatomy of its primary messenger, the neuron. While we have introduced it as the body's wiring, a closer look reveals a structure of remarkable complexity and purpose. The central component is the cell body, or soma, which serves as the neuron's life support and integration center. It houses the nucleus, which contains the cell's genetic blueprint, and is bustling with mitochondria that supply the immense energy required for constant signaling. The soma meticulously processes the myriad of signals it receives, making the critical decision of whether or not to fire an impulse of its own, acting as both a receiver and a processor in this vast network. Spreading out from the soma like the delicate branches of a tree are the dendrites. Their very name is derived from the Greek word for tree, and their function is fittingly to gather information. These numerous, shorter projections serve as the primary receiving terminals for the neuron, collecting signals from the axon terminals of other nerve cells. Their surfaces are covered with thousands of receptors, each tuned to specific chemical messengers. This intricate branching pattern dramatically increases the surface area of the neuron, allowing it to communicate with a vast number of other cells simultaneously. The dendrites funnel all this incoming information towards the soma, where the cumulative message is interpreted and assessed. Stretching away from the soma is a single, long, and slender fiber known as the axon. This structure is the neuron's output cable, designed to transmit nerve impulses over distances that can be surprisingly long, sometimes spanning over a meter in humans. The axon begins at a specialized region of the cell body called the axon hillock, which acts as the trigger zone for initiating the nerve impulse. The axon then carries this signal, undiminished in strength, to its very end. At its terminus, the axon branches into several smaller extensions called axon terminals, which are responsible for passing the signal on to the next cell in the chain, be it another neuron, a muscle fiber, or a glandular cell. Encasing the axon of many neurons is a crucial insulating layer known as the myelin sheath. This fatty substance is not a continuous coating, but is segmented, composed of individual Schwann cells in the peripheral nervous system or oligodendrocytes in the central nervous system. The myelin sheath acts much like the plastic insulation around an electrical wire, preventing the electrical signal from dissipating and dramatically increasing the speed at which it can travel. The small, uninsulated gaps between the segments of myelin are called the nodes of Ranvier. As we will see later, these nodes are vital for the rapid propagation of the nerve impulse, allowing it to jump down the axon in a process that is both swift and efficient. Before a neuron can fire a signal, it must first exist in a state of readiness, a carefully maintained state of electrical charge known as the resting membrane potential. This is not a passive state, but an active, dynamic equilibrium. Imagine a battery fully charged and waiting to power a device. The neuron at rest is precisely that, holding a store of potential energy. This potential is measured as a voltage difference across the neuron's cell membrane. Specifically, the inside of the neuron's membrane is electrically negative relative to the fluid on the outside. This small but critical electrical charge, typically measured at around -70 millivolts, is the foundation upon which all nerve signaling is built. This state of being electrically charged is referred to as being polarized. The term simply means there is a separation of opposite charges, creating an electrical gradient across the membrane. On the outside of the resting neuron, in the extracellular fluid, there is a higher concentration of positively charged ions, particularly sodium ions. Conversely, on the inside of the neuron, in the cytoplasm, there is a higher concentration of positively charged potassium ions, but also a significant number of large, negatively charged proteins and organic molecules. The presence of these large, immobile, negative molecules is a key reason why the interior of the cell maintains an overall negative charge compared to the outside. the resting membrane potential is absolutely crucial for the neuron's function. It is this pre-existing electrical gradient that provides the stored energy necessary for the rapid generation of a nerve impulse or action potential. When a stimulus arrives, it will trigger a sudden change in this potential, allowing the stored energy to be released in a controlled burst. Without this stable negative resting potential, the neuron would be unable to respond to stimuli effectively. It would be like trying to fire a slingshot without first pulling back the elastic band. The resting state is the act of pulling back that band, preparing the neuron for swift and powerful action at a moment's notice. Therefore, the resting neuron is anything but inactive. It is in a constant state of vigilance, expending a significant amount of energy to maintain its polarized state. This preparedness ensures that when a signal does need to be sent, the neuron can respond almost instantaneously, converting a stimulus into a wave of electrical activity that can travel rapidly along its axon. Understanding this baseline state is the first and most critical step in appreciating the dynamic events that follow. It is the quiet, charged silence that precedes the lightning-fast symphony of neural communication, a state of readiness that underpins every function of the nervous system. The neuron's cell membrane is the critical barrier that separates the internal and external environments, and it is this structure that makes the resting potential possible. The membrane itself is a phospholipid bilayer, a fatty structure that is naturally impermeable to charged particles like ions. Ions such as sodium, potassium, and chloride cannot simply diffuse across this lipid barrier on their own. This impermeability is what allows the cell to maintain different concentrations of ions on the inside versus the outside. However, for a neuron to function, there must be a way to control the movement of these ions across the membrane in a highly specific and regulated manner. This control is achieved through specialized proteins embedded within the cell membrane known as ion channels. These channels act as selective gates or pores, allowing specific types of ions to pass through the membrane. There are many different kinds of ion channels, but two are particularly important for establishing the resting potential. The first are the potassium leak channels. These channels are always open, but as their name suggests, they are highly selective for potassium ions. Because there is a much higher concentration of potassium inside the neuron than outside these open leak channels, allow potassium ions to follow their concentration gradient and diffuse out of the cell, leaving the inside more negative. While potassium ions are leaking out, the situation for sodium ions is quite different. The membrane has far fewer sodium leak channels, making it significantly less permeable to sodium at rest. Although there is a strong electrochemical gradient pushing sodium ions to enter the cell, driven by both the high external concentration and the negative charge inside, the scarcity of open sodium channels prevents a significant influx. This differential permeability is a cornerstone of the resting potential. The membrane is highly permeable to potassium, but only sparingly permeable to sodium. This allows for a net efflux of positive charge as more potassium leaves than sodium enters, which is a primary factor in establishing the negative interior. The selective permeability of the membrane, orchestrated by these ion channels, creates the distinct ionic imbalance that defines the resting state. The constant outward leak of positive potassium ions is the main driver behind the negative charge of the neuron's interior. Think of the membrane as a fortress wall and the ion channels as the tightly controlled gates. In the resting state, the gates for potassium are slightly ajar, allowing a slow trickle to exit, while the gates for sodium are mostly barred shut. This carefully managed flow of ions is what establishes the -70 millivolt potential, setting the stage for the dramatic events of an action potential. The constant outward leak of potassium ions through their channels is essential for establishing the resting potential, but it also presents a problem. If left unchecked, this leakage would eventually cause the concentration gradients for both sodium and potassium to run down, bringing the membrane potential to zero and rendering the neuron inactive. The cell would lose its charge, like a battery slowly dying. To counteract this constant leakage and actively maintain the crucial concentration differences, the neuron employs a remarkable molecular machine embedded in its membrane, the sodium-potassium pump, also known as Na plus K plus ATPase. This is not a passive channel. It is an active transporter that requires energy. This pump works tirelessly, using the energy derived from ATP to actively transport ions against their natural concentration gradients. For every single molecule of ATP it consumes, the sodium-potassium pump performs a specific vital exchange. It forcefully ejects 3 positively charged sodium ions from the inside of the neuron out into the extracellular fluid. In the same cycle, it captures 2 positively charged potassium ions from the outside and brings them back into the cell. This process is continuous and occurs all over the neuron's membrane, making it one of the most energy-intensive processes in the entire body. The action of the sodium-potassium pump has 2 critical effects. First, and most importantly, it maintains the steep concentration gradients for both ions. It keeps the intracellular potassium concentration high and the intracellular sodium concentration low, which is the exact opposite of the situation outside the cell. This ensures that the passive forces driving ion movement through the leak channels remain strong and consistent. Second, because the pump moves 3 positive charges out for every 2 it brings in, it contributes directly to the negativity of the cell's interior. This unequal exchange of charge makes the pump electrogenic, meaning it generates an electrical potential, further reinforcing the negative resting state. Without the relentless work of the sodium-potassium pump, the resting membrane potential would quickly dissipate and nerve function would cease entirely. It is the unsung hero of neural readiness, the powerhouse that ensures the battery remains fully charged. This active transport mechanism works in perfect concert with the passive leak channels. The channels establish the potential by allowing ions to flow down their gradients, while the pump works against this flow to restore and maintain those very gradients. Together, they create the stable, polarized state of -70 millivolts, a delicate but robust equilibrium that prepares the neuron for the critical task of transmitting information throughout the nervous system. Now that we have established the neuron's state of quiet readiness, the -70 millivolt resting potential, we can explore the electrifying event that breaks this silence, the action potential. An action potential is a rapid, temporary, and dramatic reversal of the membrane potential, a fleeting moment where the inside of the neuron becomes positive relative to the outside. This is the nerve impulse itself, the fundamental unit of information carried along the axon. It is an all-or-nothing event, a decisive signal that, once started, will travel the entire length of the neuron without losing its strength. It's the spark that ignites communication across the nervous system, enabling everything from a simple reflex to a complex thought. The initiation of an action potential begins with a stimulus. This stimulus could be a chemical signal from another neuron binding to a dendrite, or a sensory input like pressure on the skin. Whatever the source, the initial stimulus causes a small localized change in the neuron's membrane potential. It typically causes a few specialized ion channels to open, allowing a small number of positive ions, usually sodium, to leak into the cell. This influx of positive charge makes the inside of the membrane slightly less negative, a change called a graded potential. Unlike an action potential, these graded potentials are small, vary in size, and diminish as they travel away from the point of stimulation. For an action potential to occur, these small graded potentials must be strong enough, or numerous enough, to push the membrane potential at the axon hillock to a critical level known as the threshold potential. This threshold is typically around -55 millivolts. Think of the threshold as a trigger point or a point of no return. If the sum of the incoming stimuli only causes a minor change, say from -70 millivolts to -65 millivolts, the membrane will simply return to its resting state, and no impulse will be fired. This is a subthreshold stimulus. The neuron essentially ignores it, preventing the nervous system from being overstimulated by insignificant background noise. However, if the stimulus is strong enough to depolarize the membrane all the way to the -55 millivolt threshold, a complete and irreversible chain reaction is set in motion. Reaching this threshold is the key that unlocks the main event. It triggers the opening of a massive number of specialized channels called voltage-gated sodium channels, which are densely concentrated at the axon hillock. These channels are exquisitely sensitive to the membrane voltage, and the -55 millivolt level is the precise voltage that makes them snap open. This is the moment the quiet before the storm ends, and the dramatic self-propagating electrical surge of the action potential begins its journey. Once the threshold potential of minus 55 millivolts is reached at the axon hillock, the action potential begins in earnest with a phase known as depolarization. This phase is characterized by an explosive and rapid influx of positive charge into the neuron. The star players in this event are the voltage-gated sodium channels. Upon reaching the threshold, these channels instantly flip open, creating a highly permeable pathway for sodium ions, Na+, to rush into the cell. Driven by both their high concentration outside the neuron and the negative electrical charge inside, sodium ions flood into the cytoplasm in a massive, overwhelming wave. This is not a gentle leak, it is a torrent. This sudden influx of millions of positively charged sodium ions has a dramatic effect on the membrane potential. The interior of the neuron, which was negatively charged at minus 70 millivolts and then brought to minus 55 millivolts, now rapidly becomes less and less negative. The potential races past zero and actually overshoots, becoming positively charged relative to the outside. This swift change from negative to positive is the essence of depolarization. The peak of the action potential typically reaches a value of around plus 30 to plus 40 millivolts. In a fraction of a millisecond, the neuron's membrane has completely flipped its polarity, a testament to the sheer speed and power of this ionic movement. This process is a fantastic example of a positive feedback loop. The initial depolarization that reaches the threshold opens some voltage-gated sodium channels. The influx of sodium through these channels causes further depolarization of the membrane. This additional depolarization, in turn, causes even more nearby voltage-gated sodium channels to open, leading to an even greater influx of sodium. This self-reinforcing cycle creates the explosive, all-or-nothing upstroke of the action potential. It ensures that once the threshold is crossed, the depolarization process proceeds to its maximum peak without fail, generating a full-strength, reliable signal every single time. This phase is incredibly brief, lasting only about 1 millisecond. The rapid rise of the action potential is what constitutes the signal itself. It is a powerful, unambiguous on switch. For a system that needs to transmit information quickly and effectively, such as the nervous system's response to a foodborne toxin, this rapid depolarization is perfect. It creates a strong, clear signal that can propagate down the axon without weakening. The depolarization phase is the action in the action potential, the moment the neuron shouts its message down the line, initiating a cascade of communication that can have profound physiological effects. No sooner has the membrane potential reached its positive peak of around plus 30 millivolts, the process of resetting the neuron begins. This recovery phase is known as repolarization, and its purpose is to bring the membrane potential back down from its positive peak towards its negative resting state. This is just as crucial as the depolarization phase, as it allows the neuron to prepare for the next potential signal. The process involves the closing of the sodium channels and the opening of a different set of channels, the voltage-gated potassium channels. These two events happen in a beautifully coordinated sequence, ensuring a swift and efficient reset. The same depolarization that opened the sodium channels also triggered the voltage-gated potassium channels to begin opening. However, these potassium channels are significantly slower to respond. They are the more sluggish gatekeepers. So, just as the membrane potential is reaching its peak, the voltage-gated sodium channels begin to inactivate and close, shutting off the influx of sodium. At almost the same moment, the slow-to-open voltage-gated potassium channels are finally fully open. This timing is perfect. The door for incoming positive charge, Na+, shuts just as the door for outgoing positive charge, K+, opens wide. With the voltage-gated potassium channels now open, potassium ions, K+, begin to rush out of the cell. This movement is driven by two powerful forces. First, the concentration of potassium is much higher inside the cell than outside, creating a strong concentration gradient pushing it outwards. Second, the inside of the cell is now highly positive at plus 30 millivolts, which electrically repels the positively charged potassium ions, giving them an extra push to exit. This rapid efflux of positive charge from the neuron causes the membrane potential to plummet, falling from its positive peak back down into the negative territory, effectively repolarizing the membrane. This repolarization phase is the downstroke of the action potential waveform. It is the essential reset mechanism that returns the neuron's membrane to a polarized state with a negative charge on the inside and a positive charge on the outside. It is a rapid and efficient process, ensuring that the duration of the nerve impulse is kept very short. This allows neurons to fire at high frequencies when necessary, transmitting a great deal of information in a short amount of time. Without this quick repolarization, neurons would remain in a depolarized state for too long, unable to fire again, and the entire communication system would grind to a halt. The process of repolarization is designed to be swift, but sometimes it is a little too effective. The voltage-gated potassium channels that open to allow potassium to exit the cell are also slow to close. As the membrane potential falls rapidly back towards the resting level of -70 millivolts, these potassium channels remain open for a short while longer. Consequently, the efflux of potassium continues even after the resting potential has been reached. This continued exit of positive charge causes the membrane potential to briefly become even more negative than its normal resting state, a phenomenon known as hyperpolarization or the undershoot. During this hyperpolarization phase, the membrane potential might dip down to -75 millivolts or even -80 millivolts. This brief period of increased negativity is significant because it defines what is known as the refractory period. While the membrane is hyperpolarized, it is even further away from the threshold potential of -55 millivolts. This means that a much stronger stimulus than usual is required to bring the membrane back to threshold and trigger another action potential. This state makes the neuron temporarily less responsive and serves as a crucial safety mechanism, preventing the neuron from firing too rapidly and ensuring the signal travels in only one direction. The refractory period has two parts. The absolute refractory period occurs during depolarization and the first part of repolarization, when the sodium channels are either already open or inactivated. During this time, it is absolutely impossible to generate another action potential, no matter how strong the stimulus. Following this is the relative refractory period, which corresponds to the hyperpolarization phase. Here, an action potential can be generated, but only if the incoming stimulus is significantly stronger than the one that would normally be required. This period ensures that action potentials are separated events and enforces a limit on the firing rate of the neuron. Eventually, the slow voltage-gated potassium channels finally close completely, and the continuous background activity of the sodium-potassium pump takes over. The pump diligently works to restore the precise ionic balance of the resting state, moving the excess potassium that leaked out back into the cell and pumping out the sodium that rushed in. This action brings the membrane potential from its hyperpolarized state back up to the stable resting potential of -70 millivolts. The neuron is now fully reset and ready to fire another action potential. This entire cycle, depolarization, repolarization, and hyperpolarization, happens in just a few milliseconds, a testament to the incredible efficiency of neural signaling. Once an action potential is successfully generated at the axon hillock, it cannot simply remain there. Its entire purpose is to travel, to carry its message from one end of the neuron to the other. This movement is called propagation, and it functions like a self-sustaining wave of depolarization that moves sequentially down the length of the axon. The process is remarkably elegant and efficient, ensuring the signal arrives at the axon terminal just as strong as when it started. The action potential doesn't just travel like water through a pipe. It is continuously regenerated at every point along the way, preventing it from ever fading out. Imagine a line of dominoes set up in close succession. Tipping the first domino provides energy to tip the second, which tips the third, and so on, until the last domino falls. The propagation of an action potential works in a very similar fashion. The initial action potential involves a massive influx of sodium ions. These positively charged ions don't just stay in one place. They diffuse a short distance down the inside of the axon. This localized spread of positive charge depolarizes the adjacent segment to its threshold potential of -55 millivolts. This triggers the exact same sequence of events we discussed. The voltage-gated sodium channels in this new region snap open, causing a massive influx of sodium and generating a brand new full-strength action potential. This new action potential then diffuses further down the axon, triggering the next segment to reach threshold, and the process repeats over and over. This chain reaction creates a wave of depolarization that moves from the axon hillock all the way to the terminals, with each segment igniting the next in an unstoppable cascade. A critical feature is that propagation is unidirectional. The signal always travels towards the axon terminals, never backwards. This is guaranteed by the refractory period. The segment that just fired is in its absolute refractory period, meaning its sodium channels are inactivated and cannot be stimulated again briefly. While sodium ions spread in both directions, only the downstream segment is ready to fire. The upstream segment is still recovering, creating a one-way street for nerve impulse traffic. The nervous system needs rapid signaling for quick reflexes and complex thought. Myelination is the solution. Many axons are wrapped in a fatty myelin sheath formed by glial cells, which acts as an electrical insulator. Myelin prevents ion leakage, allowing electrical signals to travel farther and faster down the axon. In unmyelinated axons, action potentials must be regenerated at every point, slowing transmission. Myelinated axons allow signals to skip insulated segments, drastically increasing speed. This adaptation enables high-speed communication, crucial for fast reactions and advanced nervous systems. Myelin is a key evolutionary advancement, making rapid, complex responses possible. Myelin sheaths are segmented, with gaps called nodes of Ranvier. These nodes are packed with voltage-gated channels, while the myelinated segments have few. Action potentials leap from node to node, a process called saltatory conduction. The signal travels rapidly through myelinated segments, regenerating only at the nodes. This leapfrogging makes conduction much faster than in unmyelinated axons. Saltatory conduction is not only fast, but energy efficient, requiring less work from the sodium-potassium pump. Myelinated axons can transmit impulses up to 100 meters per second. This efficiency is vital for rapid, high-priority communication in the body. Nerve impulse speed depends on myelination and axon diameter. Myelinated axons transmit signals much faster, especially when combined with a large diameter. Wider axons have lower resistance, allowing current to travel more quickly. The fastest neurons are both myelinated and wide. Superhighways for urgent signals. Slow, narrow, unmyelinated axons handle less urgent information, like dull pain or body temperature. This diversity lets the nervous system allocate resources efficiently. Myelination and diameter together explain the wide range of signaling speeds in the body. The all-or-none principle means a neuron fires a full action potential if threshold is reached, otherwise, nothing happens. There's no such thing as a weak or partial action potential. This ensures reliable, uniform signals across long distances. The axon hillock acts as the trigger once threshold is crossed. The process is automatic. Every action potential is identical, preserving message strength from start to finish. This digital on-or-off system prevents signal degradation and confusion. Like binary code, the neuron's firing or not firing is clear and robust. This principle is essential for accurate, unambiguous communication in the nervous system. If every action potential is the same size, how does the nervous system signal intensity? The answer: frequency. Stronger stimuli cause neurons to fire more rapidly. A light touch triggers a slow firing rate. Intense pressure triggers rapid volleys of action potentials. The brain interprets high-frequency signals as stronger stimuli. It's like Morse code. The message is the same, but urgency is shown by speed. This frequency coding is universal, letting us distinguish between mild and intense sensations. For food safety, a powerful toxin triggers a rapid, urgent signal, alerting the body to danger. Fast, high-frequency nerve signaling is crucial in responding to foodborne toxins. Toxins in contaminated foods stimulate sensory nerves in the gut, sending rapid signals to the brain's vomiting center. Myelinated fibers ensure this warning travels almost instantly, triggering vomiting to expel toxins. Some pathogens, like Clostridium botulinum, produce neurotoxins that block nerve signals, causing paralysis. Understanding nerve conduction highlights the real dangers of foodborne illness. Failures in food safety can disrupt the body's most vital communication systems. Rapid symptom onset in food poisoning shows just how efficient and vulnerable our nervous system is. As a food safety officer, knowing this physiology underscores the importance of your role. As a food safety officer, your role is to stand as a barrier between the public and the potential hazards lurking in the food supply. While you might not be diagnosing patients directly, understanding the physiological consequences of contamination really transforms your job from just a checklist of tasks into a mission of public health protection. When you inspect a facility for proper temperature control, you're not just checking a number on a thermometer, you're actually preventing the growth of bacteria like Listeria or Salmonella that can trigger violent nerve-mediated responses like diarrhea and fever. You are, in a real way, safeguarding the delicate electrochemical balance of millions of neurons in every potential consumer. Consider for a moment the sensation of taste and smell, which are honestly our first lines of sensory defense against spoiled food. These senses are entirely dependent on nerve impulses. When food starts to spoil, it produces chemical compounds that our olfactory and gustatory neurons recognize as off. These neurons fire action potentials to the brain, creating the perception of a foul smell or a sour taste, which triggers an aversion response. This is the nervous system at work, providing an early warning. Your job in enforcing food quality standards helps make sure that food never reaches the point where a consumer's own nervous system has to make that last-ditch defensive judgment. Moreover, knowledge of how the nervous system functions gives you insight into the range and severity of symptoms associated with different foodborne illnesses. Some pathogens cause localized gut irritation, while others, like certain types of seafood toxins, such as ciguatoxin, can cause bizarre neurological symptoms like temperature reversal sensation, where hot things feel cold and vice versa. This happens because the toxin directly interferes with the function of voltage-gated sodium channels, disrupting the normal pattern of action potentials. Understanding the underlying mechanism really helps you appreciate the diverse and serious threats you are working to mitigate. Ultimately, every regulation you enforce, from hand-washing protocols to cross-contamination prevention, is designed to keep the human body's internal environment stable and free from attack. The nervous system, with its lightning-fast action potentials and all-or-none signals, is the ultimate sensor and responder to these attacks. By grasping the fundamentals of how this system works, you gain a much deeper respect for the biological systems you protect. Your work ensures that the only signals traveling through a person's nervous system after a meal are those of satisfaction and nourishment, not the frantic high-frequency alarm bells of a body under siege from a preventable foodborne threat. Alright, we have journeyed through the intricate world of the neuron, from its quiet resting state to the explosive firing of an action potential. We have seen how this signal travels, leaps, and carries vital information throughout the body. Now it is time to consolidate that knowledge and test your understanding of these core concepts. This short quiz is designed to reinforce the key points we have covered. It is a chance for you to pause, reflect, and see how well you have grasped the fundamental mechanisms of nerve impulse conduction. Don't worry about getting everything perfect. The goal is to identify areas you understand well and those that might need another look. Let's begin with the first question. First, what is the name for the stable, negative electrical charge across a neuron's membrane when it is not actively firing a signal? The answer is the resting membrane potential. This state, typically around -70 millivolts, is the baseline of readiness, maintained by the constant work of the sodium-potassium pump and the differential permeability of the membrane to key ions like potassium and sodium. It is the charged potential that makes the subsequent action potential possible. Thinking back, which pump is essential for maintaining this gradient by actively moving 3 sodium ions out for every 2 potassium ions it brings in? The answer is, of course, the sodium-potassium pump. Next, let's focus on the action potential itself. Which specific type of positive ion floods into the neuron to cause the rapid depolarization phase, flipping the membrane potential from negative to positive? The correct answer is sodium ions, or Na+. This influx is triggered when the neuron reaches its threshold potential, causing voltage-gated sodium channels to open. Following this depolarization, what is the process called that restores the negative charge inside the neuron, primarily caused by the exit of potassium ions? This crucial reset phase is known as repolarization, which ensures the neuron can fire again. It's the downstroke after the spike. Finally, let's review the structures that enable high-speed communication. What is the name of the fatty insulating layer that wraps around many axons, dramatically increasing the speed of nerve impulse transmission? That would be the myelin sheath. And what do we call the remarkable process where the nerve impulse jumps from one gap in this sheath to the next? This rapid form of propagation is called saltatory conduction. And what is the fundamental principle stating that a neuron either fires a full-strength impulse or none at all? That is the all or none principle, which ensures the signal is a clear, unambiguous digital message. Reviewing these concepts is more than just an academic exercise. It is about building a solid foundation of physiological knowledge that directly supports your future career. The journey of a nerve impulse, from a resting state to a propagated action potential, is a story of precision, speed, and efficiency. Understanding the resting membrane potential reveals how our bodies invest a tremendous amount of energy simply to be ready to respond at a moment's notice. This state of readiness, maintained by the diligent sodium-potassium pump, is the silent sentinel guarding against delayed reactions, ensuring the nervous system can spring into action instantly when a threat is detected. The action potential itself, the dramatic sequence of depolarization and repolarization, is the language of the nervous system. The all-or-none principle ensures this language is clear and decisive. There are no whispers or mumbles. There is only a clear yes or no, a fire or don't fire. This digital clarity is essential for a system that must make life or death decisions. The way the nervous system encodes the intensity of a stimulus, not by changing the size of the signal, but by altering its frequency, is a brilliant solution that allows for a nuanced understanding of the world while maintaining the integrity of the message. A high-frequency signal is an undeniable scream for attention. Furthermore, the evolution of the myelin sheath and saltatory conduction underscores the body's relentless drive for efficiency and speed. This biological innovation allows for near-instantaneous communication between distant parts of the body, enabling rapid reflexes and complex processing. For a food safety officer, this translates into understanding why the symptoms of certain foodborne illnesses can appear so quickly and dramatically. The body's alarm system doesn't send a letter. It sends a high-speed digital alert via these myelinated superhighways, triggering protective responses like vomiting or diarrhea to expel a threat before it causes greater harm. Ultimately, every stage of nerve impulse conduction has real-world relevance to your field. From the initial sensory neuron detecting a toxin to the motor neuron that is disabled by a neurotoxin like botulinum, the principles we have discussed are at play. Your role in preventing contamination is a role in protecting this intricate and vital communication network. By ensuring the safety of the food supply, you are making sure that the only signals being fired are those of healthy bodily function, not the frantic, high-frequency warnings of a system under attack from a preventable pathogen or toxin. This knowledge connects your daily duties to the profound well-being of the public you serve. We have traveled an incredible distance in a short time, moving from the basic structure of a single neuron to the complex, high-speed symphony of a propagated nerve impulse. We started with the quiet, charged readiness of the resting membrane potential, a state of constant vigilance powered by the tireless sodium-potassium pump. We then ignited the spark of the action potential, witnessing the explosive influx of sodium during depolarization and the swift reset of repolarization. This journey has shown you that the nervous system doesn't just send messages, it crafts them with incredible precision, using a digital all-or-none language to ensure every signal is clear, strong, and unambiguous from start to finish. This exploration has equipped you with a new lens through which to view your future profession. The concepts of threshold potential, saltatory conduction, and frequency coding are not just abstract biological terms. They are the underlying mechanics of the very systems you will be protecting. You now understand how a signal is born, how it gathers speed by leaping down a myelinated axon, and how its urgency is communicated through a rapid-fire volley of impulses. This knowledge transforms the idea of a symptom from a simple observation into a tangible physiological event, a direct consequence of the body's nervous system reacting to a threat. This journey from a microscopic ion channel to a full-body reflex is a testament to the elegance and efficiency of our biology. It's a system honed by evolution to perceive the world, coordinate action, and most importantly, defend itself from harm. As you move forward in your studies and into your career, carry this understanding with you. Let it serve as a constant reminder of the intricate and delicate biological machinery that is at stake. Your work is not just about enforcing codes and regulations. It is about protecting the very electrical grid that powers human life, ensuring it runs smoothly and without interruption from preventable hazards. We encourage you to feel confident in this new layer of understanding. You have successfully navigated the fundamental principles of one of the most complex systems in the human body. This knowledge doesn't just make you a better student, it makes you a more informed and insightful future professional. It provides a deeper why for the what of your daily tasks. By grasping how the body communicates danger, you are better prepared to recognize the importance of preventing that danger in the first place, reinforcing your critical role in the vast network of public health. Ultimately, our deep dive into the conduction of nerve impulses circles back to one central theme: the profound connection between food safety and human physiology. Your future role as a food safety officer places you at a critical intersection, where public health policy meets the intricate workings of the human body. Every standard you uphold and every inspection you conduct is a direct action to maintain the body's natural harmony and prevent the chaos that foodborne pathogens and toxins can unleash upon the nervous system. You are, in a very real sense, a guardian of physiological stability on a community-wide scale. Think of the all-or-none principle. A consumer cannot be a little bit sick from a powerful toxin. The body's alarm systems, once triggered, will launch a full-scale response. Your diligence ensures that the stimuli that could trigger these alarms are eliminated from the food supply chain. When you enforce rules about proper cooking temperatures, you are not just following a guideline. You are destroying bacteria that could release toxins designed to disrupt the very ion channels we have discussed, preventing a cascade of frantic, high-frequency signals that lead to illness. Your work happens upstream, so the body's own defense systems don't have to engage downstream. This knowledge should empower you. It provides a solid scientific foundation for the importance of your work. You are protecting more than just people from getting sick. You are protecting the integrity of their neural pathways, the precision of their neuromuscular junctions, and the stability of their internal environment. When you prevent an outbreak, you are ensuring that thousands of nervous systems continue their normal, essential work of managing thoughts, movements, and sensations without being hijacked by an invading microbe. It is a profound responsibility, and understanding the science behind it only makes your purpose clearer and more compelling. So, as you conclude this lesson, take a moment to appreciate the bigger picture. The journey of a nerve impulse, from potential to action, from axon hillock to axon terminal, is a microscopic marvel that enables life as we know it. Your career will be dedicated to protecting this marvel in countless individuals you may never meet. We hope this class has not only demystified the nervous system, but has also fortified your commitment to your chosen field. Thank you for joining us on this exploration, and we look forward to seeing you continue to build the knowledge that will make you an exceptional food safety officer.